Now I'm recording. Okay. So first, we'll focus on the bacterial actions and then how that relates to plants. And I like to start with this picture, which did not come from your book. I didn't like the pictures that were in the book, so I went and got this one off of the internet. And this is showing several different nitrogen relationships amongst organisms. So it's showing how do different plants relate to nitrogen, how do different bacteria process nitrogen in different ways down here. We've got decomposers that are kind of recycling nitrogen out of plants and back into the soil. And then the rabbit, which this is related to the rabbit as well, just because the rabbit eats the plants. So this particular picture is going to be the board or the screen picture for the next exam. To kind of jog your memory about what are the different relationships between these organisms. And you'll have, several short answer or discussion questions about this system. So this will be the board picture. There'll be several questions about it. And I'll come back to this kind of toward the end of lecture to talk about how this relates to what we've learned about the individual parts. That will be the big focus exam picture. There we go. Okay, so I want to start with why nitrogen fixation is so important. And when I was putting together this lecture several years ago, I found this flyer from the 10th annual European Nitrogen Fixation Conference. So there are enough people that study nitrogen fixation that they have conferences about it and they have whole journals about it and share their research, and it's a big deal in plant sciences. And this is why. So all life needs elemental nitrogen, like some form of nitrogen element, for what? What are the major molecules that we make that have to have nitrogen in them somewhere? And then biochem folks have been drawing some structures or memorizing some structures that have nitrogen in the center of them. The amino acids, definitely. So you can't make any amino acid without a nitrogen central atom to build that around. So you have to have them for amino acids, and therefore you have to have nitrogen for protein. And that's one of the two huge uses of nitrogen for life. Every amino acid has to have nitrogen, and every protein has to have multiple amino acids. And then the second one that all life has to have is there is quite a lot of nitrogen in nucleic acid bases. So they're called nitrogenous bases. Those contain nitrogen as well. So that's like 2 out of the 4 major macromolecules are absolutely dependent on nitrogen. And a lot of let's say one of the limiting factors for life in an ecological sense is, does it have adequate nitrogen supply? So fixing nitrogen, if we talk about fixing nitrogen, what that really is taking nitrogen gas from the air and getting it into a usable nitrogen form to begin the process of forming these two. All life has to have nitrogen for its amino acids and for its nucleic acid bases. And the greatest source of nitrogen on Earth is in the air that we breathe. Nitrogen gas. One of the molecules I can still draw and get it correct. There you go, nitrogen gas. N, triple bond in between. That is the dominant nitrogen source on Earth. And if organisms have the ability to do this, to take nitrogen out of the air and turn it into something that can begin to be a building block for these, that is a huge advantage. That's what nitrogen fixation is. Before I go on into that, carbon fixation is a similar process to pull CO2 out of the air into a more usable organic form of carbon. So when we talk about so-and-so fixation, it's grabbing the basic element out of the air and turning it into a more usable form. Which one has more in the air? That's lame. Which one is a greater percentage of air, nitrogen or CO2? You may not have talked about this since like middle school in a sentence. Nitrogen is 70-some% of the air into gas. And then carbon dioxide is around like 2 or something like that. And then oxygen is either 19 or 21, something like that. So there's a lot of nitrogen in the air. We breathe it in and we breathe it back out, and it just goes, it never changes by our metabolism. But there's certain microbes that can fix nitrogen and make it available for all life. So we're used to looking at plants in an ecological sense as primary producers. Like they're the primary producers and they're doing a lot of carbon fixation, and then other organisms come along and eat them, and they're the bottom of the food web, or that's not a good way. They're the beginning of the food web as primary producers. But plants oftentimes are totally dependent on fixed nitrogen in the soil that come from bacteria. So that's the last fact that I want to write about why nitrogen fixation is important. Plants are primary producers in a carbon sense. Like they are the carbon base of the food web. But plants themselves are dependent, or they depend on fixed soil nitrogen. And all of the fixed soil nitrogen, almost all of it, is coming from bacteria. So without nitrogen fixation, you can't have primary producers in a plant sense. So any organism that eats plants, including us, is dependent on nitrogen fixation by bacteria in the soil. And different groups of bacteria are doing different nitrogen processes. They kind of interact ecologically and depend on each other. So this is the equation for nitrogen fixation, and it's kind of basic. So we start with nitrogen gas in the atmosphere. There's an enzyme called nitrogenase that converts nitrogen gas into ammonia. So starting with N2, there's an enzyme called nitrogenase that can turn it into ammonia, and I bet you remember that chemical formula already. The other remarkable part of this is how energetically expensive it is. A 12 ATP reaction is super expensive. But this is so valuable to fix nitrogen that it's worth it for organisms to spend that much ATP to pull usable nitrogen out of the air. So, number one, not all bacteria can do this. Not all bacteria can do it, but the ones who can end up producing a lot of usable nitrogen for the whole rest of life on Earth. They get the process started. And the reason it's so energetically expensive is that nitrogen is a triple, N2 gas is a triple covalent bond that has to be broken to be converted into ammonia. So those are kind of the basic facts about the equation. So nitrogenase as the enzyme, it takes 12 ATPs to end up with 2 ammonia. Then ammonia, NH3, will later on eventually get converted to nitrites or nitrates by other organisms. So the first step is just to get ammonia. Nitrogen gas is hard to use, hard to work with. Ammonia is much easier to work with. It's much more reactive. Nitrogen gas is very stable, totally triple covalent bonded, very hard to start working with, and nitrogenase is the key enzyme to do this. Only some bacteria have nitrogenase. Nitrogenase enzyme, could you turn off one of the other light? Nitrogenase enzyme is a big multi-component complex. It shows quaternary structure. And it's basically symmetrical with one part that's called dinitrogenase, like N2 breaker. Dinitrogenase. And then a sort of a companion domain called dinitrogenase reductase. Details of that, I don't need you to so much know, but the whole thing together is referred to nitrogenase like as a complex. So that's like calling, if you turn the lights back on, one light. Like we talk about ribosomes, and ribosome is an enzyme complex that's actually got multiple parts. Nitrogenase is a complex that has multiple parts. For organisms that have to have, or that can have nitrogenase and can fix nitrogen, they require this cofactor called FMOCO, is the short name for it. And this is showing the whole entire cofactor. But what's important to note about this is that it requires iron. So there has to be a good iron supply. As you see, I guess there's seven iron atoms in this cofactor. There's also molybdenum. Do you ever talk about molybdenum after like periodic table time? I don't think I did in chemistry. This is a place where molybdenum, can you say it? Molybdenum is super important. And what's the co? what's that element, CO? Cobalt. Which in human metabolism, like we need cobalt, but just barely, and we never talk about it. But those three elements are essential to nitrogenase activity. And bacteria that can do nitrogen fixation would be dependent on these in their environment to keep doing it. I think this one right here is the cobalt that's not labeled. But the whole nitrogenase complex requires FMO cofactors in those positions. Something interesting about nitrogenase is that if it's in the presence of oxygen gas, so up here where it says oxygen, like put up above that O2, not just any oxygen atom, but oxygen gas. In aerobic conditions where oxygen gas is present, that inhibits nitrogenase, which kind of suggests that nitrogenase is most likely more ancient than oxic world. Nitrogenase developed so long ago, before there was oxygen on Earth. And then once oxygen gas was in the atmosphere on Earth, organisms had to adapt if they were going to keep using nitrogenase to keep oxygen away. So many of the bacteria that use nitrogenase and fix oxygen will have some specialized slime layers that can exclude oxygen from diffusing in, which is kind of unusual and weird. So this layer right here, I think this is part of the quiz, that's going to prevent oxygen gas outside the cell from diffusing inside the cell. And I've never looked up like how the slime layers do that, but it's a cool concept. They prevent oxygen from messing up the activity of nitrogenase. This slide I want to skip over. Just cross that out. Scientists who study nitrogen fixation kind of fall into two groups. About half of them are interested actually in bacteria that do nitrogen formation. So they would be more on the microbiology side. And then the other group of scientists that massively care about this are agricultural plant science people. And that's kind of the world that I did my undergraduate in. And then when I got into my PhD program, I encountered some of the people who are more like microbiology focused on this. But to study nitrogenase activity is not that easy, because you got to keep it away from oxygen. And if you're trying to measure production of nitrogen gas, and like 80% of our air is nitrogen gas, it's kind of rough. So the way that scientists actually study nitrogenase activity in human air is that nitrogenase can actually turn acetylene, like blowtorch acetylene, into ethylene. And that's how they measure its activity, instead of trying to say, oh, well, we detected that the air went from 79% nitrogen to 80% nitrogen. That's too error prone. So it's tricky to work with nitrogenase experiments, but it's so important that people find a way to do that. Okay, so now we have a totally different picture. And this is something I want you to understand, concepts, not details. So not all the bacteria can fix nitrogen, but the ones that do, it requires a pretty big set of genes to make nitrogenase and all the other things that are required. So this is a map of how many genes would it take for one organism to do all the stuff it needs to fix nitrogen. And so we would call that the nitrogen fixation regulon. So it's multiple genes within the genome, and they're all kind of controlled as a set. So abbreviation is NIF regulon. So you have to have genes to be able to process molybdenum. Then you have to have genes to build FOMOCO enzyme. You have to have some regulatory and sensing genes. This is a part of the larger enzyme. Some more genes to build FOMOCO. Some more genes to build FOMOCO. A lot of genes to put together nitrogenase. And that's why not so many bacteria can do it, because you would have to have the whole set. Instead of just picking up like, oh, in transformation, oh, I've got a capsule gene now, I can do it. You have to have all of this set. And this, the reason that we know so much about this is that plant scientists would love to be able to make plants that just do this on their own. That would be awesome. Like, plant a plant in crappy soil that has low nitrogen content, low fertility. If you could engineer it to fix nitrogen on its own, that would be amazing for agriculture and food production. But it turns out that's a really tall order, because this is so many genes to introduce into a plant. So that's not going to happen anytime soon. Only certain bacteria can do this, and as a group, these are called proteobacteria. So I'm going to write that word here, but really it should be up here. Proteobacteria. And together as a group, all of these are gram-negative. So we've got here all these sets of bacteria, and some of them are labeled alpha, some of them beta, some of them gamma. Together, all of them are called proteobacteria. And proteobacteria happen to all be gram-negatives. So which one up here have you ever seen before? Pseudomonas. Yeah. So the Pseudomonas family of bacteria are able to fix nitrogen. All of these others, there's one other that can cause a human disease, which is gonorrhea. Interesting. It's Neisseria family. But all of these others tend to never be talked about in medical microbiology, because these are mostly soil related and plant related.
What's the Bordetella one?
Oh yeah, Bordetella, like, thank you. Bordetella pertussis, like whooping cough. That's another one I forgot about that is human related. So this one, and let me double check. There's also, Kate and Emily, one called Brucella abortus, which causes, it can cause infections in cows and cause them to lose the calf. Brucella abortus. And they pick it up from soil when they're eating their grass, grazing. That's what I mean. And then this one, Rickettsia family, those can cause either Lyme disease or Rocky Mountain spotted fever. that set can fix nitrogen. And if you can fix nitrogen, you're gonna have an advantage to the bacteria around you that can't, because you're gonna have a little bit more availability to build amino acids and to build nucleic acids. Okay, we're gonna have our break right now because the next section then turns to plants. So that's the section about bacteria, plants, and the symbioses between them are coming next. No, it'll need to be down the wall. So like at 10, that would be good. I'll have all the stuff set up. Yeah. Yeah, I forgot to mention that, but it's easier to move ourselves to there than bring all that here. And on Thursdays, I don't have the A&P labs over here, so. Kate, how have you learned about Bordetella before?
That's one of the things that can cause like kennel cough and
Yeah, kennel cough. It's whooping cough, but it's cough. Yeah. Yeah, so you just learned that working?
yeah.
Yeah, I don't know as much about the animal diseases. I never, like, if here's a new thought, can pets get STDs?
Yes, I know cats can.
Cats can.
Yes.
Never thought about that before. I have like human focus and then I have bacteria focus and then all the other life forms are kind of like
Yeah.
afterthoughts.
HIV.
That's right. Isn't it like FIV?
Yeah.
Yeah. Feline. Yeah. I've been watching, there's a new PBS remake of All Creatures Great and Small. And when I was young, I read all of those books. I was crazy about them. And then I watched the series and it's so good, but it only had like 7 episodes. So I went back to the books and started reading them. And the books mention a lot of these different infections, which I had forgotten. Xanthomonas is one of the major plant pathogens. So when I was in graduate school, there was a lot of research about Xanthomonas plant infections. Okay, so before I get into the plant part, I want to go back to this picture and relate nitrogen fixation that we just talked about to what's going on in the soil here. So in this picture, like everything from here down is considered in the soil. And then you've got plants growing above the soil. So there are actually 3 different types of nitrogen conversions that different bacterial groups can do. And everything I just said previously is about the nitrogen fixing bacteria. So those are shown here, that some of those can be in association with plants, but they also can live just in the soil. So that first category that do nitrogen fixation, they're converting into ammonia. That's what nitrogen fixation is. And those can be either just in the soil, like they live in the soil, and we call that free living. Just kind of hanging out in the soil and living their life that way. Or they can be in nodules that are symbiotic with plants. There's more of them that are free living than are with the plants in nodules. So that's the first group. And the other groups are not going to be able to do anything without the action of nitrogen fixers. So they're kind of at the beginning of all of these nitrogen conversions. There's another group of bacteria that are called nitrifying. That's group 2, nitrifying bacteria. And the nitrifying bacteria are doing one of 2 things. So they're either taking ammonium ion. So ammonia and ammonium ion, those will interconvert states depending on the soil pH. They're basically the same thing. Ooh, don't tell Dr. P I said that. But like biologically, they're pretty similar. They're just with or without an extra proton. So nitrifying bacteria can either take ammonium and convert it to nitrites, and that's usually going to be the first step. So they can do ammonium to nitrites, or then converting nitrites to nitrates. Both of those processes are considered to be nitrifying. And the reason that they got that name, nitrifying, is that nitrate, NO3-, nitrate ion, is that what you call it? Is it just an ion, or is there another name for that? Nitrate, like this. That's the actual usable form of nitrogen for plants. Plants uptake nitrate. They don't use nitrogen gas, they don't use nitrites. They are equipped to intake nitrates up through their roots. And so that's their nitrogen feeding strategy. So nitrifying bacteria are taking ammonia or ammonium and pushing it in the direction of availability to plants. And so plants like to be in the environment of nitrogen fixers and nitrifying bacteria. The third class of bacteria is doing something completely different, and they're called denitrifying. And they're basically the opposite of group 2. They're shown over here. The denitrifying bacteria are taking nitrates, which is what plants like, and they're turning it back into nitrogen gas. They're making nitrogen unavailable to plants, and they are basically unfertilizing the soil, is the way that you can think of it in a plant-centric way. This is what plants need. There's a group of bacteria that take nitrates and convert it back into nitrogen gas, which is useless to plants. So the denitrifying bacteria are doing this. And if you farm or, are in agriculture industry, denitrifying bacteria are basically your enemy. Like, you would not want them to be around. And the denitrifying group denitrifying soil bacteria are always there. There may be more or less in a certain soil. And denitrifiers tend to thrive in anoxic conditions in the soil, and especially flooding. When the soil gets flooded with water, like in a big flood, I used to think that the main problem with flooding is that it's just too watery. It's just too watery down there, and the water is going to kill the plants. That's not actually the main problem, because the water will kind of get soaked up by the plants, but the water is crowding out oxygen in the soil. So the roots don't get enough oxygen, and all the time while the water is there flooding out oxygen, the denitrifiers tend to get going. And the longer the soil stays flooded, the more nitrates are going to be lost out of the soil, and the soil becomes less fertile or less beneficial for the plants the longer it's flooded. So denitrifiers are bad news for plants. Final thing on this before we go to the nodulation is that the step in between ammonia and ammonium, that's called ammonification. That's more dependent on soil pH than microbe action. But right here where they're saying ammonification, it's technically ammonia that's produced during nitrogen fixation, and it's ammonium that is the basis for nitrifying. So that's what this ammonification word is there. So plants are always going to benefit from nitrogen fixing bacteria, even if they're just in the soil and not together with the plant. And plants are going to benefit from nitrifying bacteria as well. So both of those make for healthy soil for growing plants in. But if the plant has right there with it, right with its roots, nitrogen fixing bacteria in symbiosis, they're going to do a lot better than plants that don't have the nitrogen fixing bacteria in symbiosis. So certain plants are able to form symbiotic association with nitrogen fixing bacteria, and there's a cooperation between the plant and the bacteria. They make these little incubators called nodules. Not all plants can harbor nitrogen fixing bacteria. and form nodules. So it's special. The plants that can do it are mostly what we know as legumes. The leguminous plants. And they're put into the legume group because they can do this. If you've got nodules and bacteria that fix nitrogen in your vicinity, you can do better in poor soil because it's like you've got your own little fertilizer factory down there at your roots, giving you more nitrogen. So Jamie, could you turn one light again? The plants on the left have been cured of their nodules. Is that weird to think of? This is technically an infection. The plants are infected in a way with these nitrogen-fixing bacteria. These are regular soybeans, which are legumes, which make nodules and do the symbiosis. If you cure them of the infection with the nitrogen-fixing bacteria, they grow like this. And they look like they need fertilizer, right? Same plants, infected or symbiosing, not infected. So that shows you the advantage of being able to form symbiotic nodules. What are, turn one light back on. What are all of the legumes that you can think of? Soybeans the first. What else? What other plants make high protein that we like to eat? Peas, yeah. So soybeans are, you could write some on here. Soybeans, peas, peanuts are. And then alfalfa, which most people don't care about, but it's great nutrition for animals. Alfalfa is a legume. It's also one of the spelling bee words I missed in the year I didn't win the championship. It's not even that hard. Alfalfa. It's exactly like it looks. So soybeans, peas, peanuts, alfalfa. And there's probably a couple others, but those are the good ones to know. And for people, especially if you're vegetarian or vegan, and you get your protein from plant sources, you're really dependent on legumes for your protein supply. For your tofu, your peanut butter, lentils, which are kind of pea. Those, because they've got nodules, they fix a lot more nitrogen, and the seeds that they make that we steal from them also are going to have high protein content. Plants that can do nodulation and support symbiosis of the nitrogen fixers, they have to make a good home for the bacteria if this relationship's going to work out. So they've got to have a way to keep oxygen away from the bacteria. And the way that plants help with this is they can make a version of hemoglobin, like hemoglobin that's in our blood that binds oxygen and carries it around. Some plants can make a version of hemoglobin called legumoglobin, like from legume, legumoglobin, and it binds oxygen really tightly where the nodules are and keeps it away from the bacteria. You might think, what in the world is that? These are like a plant embryonic tissue that is showing legumoglobin kind of in red. So you could take this embryonic tissue and form a whole new plant out of it by tissue culture. But the book I used to teach out of, this is how they show legumoglobin. So the plants have to make low oxygen conditions for the bacteria. But to really get nodules going, you have to have basically an infection. So nodules are going to be basically on the roots, under the soil, and they're a little home or special incubating chamber for the nitrogen-fixing bacteria. When you plant a seed of a legume, it does not have nodules with it. So the plant will grow, and every new plant has to get infected with the bacteria that can symbiose. And this is how that works. The whole process of that is called nodulation. Nodulation is the process of establishing the symbiosis. It requires a lot of specific things to happen. But all of this together would be called nodulation. And it's a type of infection that stays, and it benefits both organisms. So we're going to start here. This is a plant root, and it's got root hairs, which are tiny extensions off of a single cell. So they're drawing a box around the root hair. And this blue right here, that's the end of a root hair, kind of like growing out into the soil. And it's got receptors on its surface that can bind onto a nitrogen fixer. So what they're calling here rhizobial, that's a nitrogen fixing bacterium. This red that they're calling rhizobial cell is nitrogen fixing bacteria. And there has to be a specific binding recognition there. Plants will not reach out to an E. coli cell they won't do this for an E. coli cell. It's got the wrong shape. There's a recognition and attachment. And then once you have this binding event, so you could write here binding. Once the bacteria sense the binding, they are going to make this chemical called a nod factor. Nod factor. So nod factors are made by the bacteria, and they are diffusible chemicals that are going to encourage the root hair to curl. This is really cool. So first, the root hair and the bacteria bind. The bacteria sense that they're bound to a root. And then they make these chemicals called nod factors and release them into the area. So here's a picture of a nod factor. Kind of looks like that. So it's kind of a long, it's a pretty big molecule. But that particular chemical is sensed by the plant, and the root hair, when it senses that, it's going to curl, and it curls around the bacteria. So this is maybe stupid, but so if this is the bacteria and I'm the root hair, and they touch, and then you can imagine like chemicals coming out of the marker, and then I'm going to curl and like engulf it, almost swallow it, surround it. And there's kind of like a little nest in there. And then after that, the bacteria are going to kind of start dividing in there. So they multiply within the curl. And then as they multiply, they're going to start going into the plant. They form what's called infection thread. So they're dividing and kind of migrating in this direction. And the infection thread will kind of infiltrate the root and grow into the root. And then from there, the plant root cells are going to divide like they have cancer. So much going on. Like curl, infection thread. And then when the plant senses that the infection thread is in the root is going to, root cells are going to divide kind of like cancer and form something that's kind of like a tumor for the bacteria to live in. So this next picture shows that a little bit better. Jamie, could you turn the lights off again? It's a busy day for you. Thanks. Okay, so this is a picture of root hair. So it's in this plane and then it's kind of curling around to this plane. And it has already had started in it the infection thread. So these little glowing ovals, which are gram-negative rods, by the way, they're making the infection thread and they're heading into the root. And then after maybe 3 days, once the infection thread is kind of right here, they've encouraged the root cells to form this tumor-like thing. So this is the pre-nodule, and the bacteria that are in there kind of shut down some genes and they become less independent, and they turn into something called bacteroids. So, Jamie, if you could turn back on one light. So the bacteria inhabiting nodules are going to adjust their gene expression. And they become more dependent on the plant. And when they're doing that, they're entering this state called bacteroid. You're kind of less than a fully competent, free-living bacterium. You're bacteroid. You're dependent on the plant, and then the plant's going to be kind of dependent on you. So the bacteroid stuff is happening between infection thread and having full-on nodules. And nodules are kind of like a cancer that's good for you if you're a plant. When the plant dies, like the bacteroids they're kind of going to die too. But there's other bacteria out here in the soil that could colonize the next plant. So the new plant will have to recruit new bacteria, curl the root hair after they get the nod factor, form infection thread, form the bacteroids and the nodules, and that repeats with every new plant. This process is really well worked out, and probably thousands of plant scientists and microbiologists have worked out a lot of detail about all of this, because it's so, if you could figure out the secrets and get plants to do it for themselves, you could feed so many more people. Or you could grow plants in soil that right now is worthless. So it may sound dorky, but it's like this is a huge deal in plant science and agriculture, and there are so many labs that work on this kind of thing. Some of them, like field agriculture, and some of them, like the people never leave the lab, but they're doing agricultural-based science. I kind of dork out about this because that's the world I came out of. This slide we're going to skip over. Like this is another version of nod factor and anti-nod factor. So there are certain chemicals that prevent nodulation and inhibit them. But I'm not going to say a whole lot more about that. This picture is kind of showing what do plants and bacteria get out of this? Like, what's the benefit of the symbiosis for both parties? So, this is an ecology year. What is the name of this kind of symbiosis where both of them get something good? Mutualism. Yeah, this is mutualism. So you write that right here, mutualism. And what I want to mostly focus on in this, so this is the bacteroid. It's still kind of like a gram-negative rod-shaped cell. And then out here is the plant. What is the plant getting out of this deal that's beneficial? What do plants need from these nitrogen-fixing bacteria? Yeah, usable nitrogen. And this is, like what it's showing here is not something I have mentioned yet, but here's the nitrogenase turning nitrogen gas into ammonia. And then it's showing ammonia is being exported to the plant as glutamine and asparagine. What are those? Those are amino acids. So in this scenario, the plant is actually directly getting amino acids from the bacteria that live with it. And those also are a form of usable nitrogen. And the plant can turn these into other nitrogen compounds that it needs. So in the symbiosis, the plant is directly getting amino acids already. Like, it doesn't even have to convert nitrate to amino acids, it's getting amino acids specifically. What is the bacterium, or the bacteroid, what does the bacteroid get out of this deal? A better place to, yeah, it in some ways gets protection, like by living in the plant root. It's protected from other tiny organisms that would eat it in the soil. What else is it getting chemically out of this? Yeah, it gets ATP. And part of the ATP is going to use to run nitrogenase, but it's getting basically food. So we're showing here organic acids. That's going to be its major carbon source and its major fuel is the organic acids. And these are the specific ones that it's most interested in having. So succinate, malate, fumarate, those can power citric acid cycle and then eventually power electron transport chain. And these are like instead of sugars. So like we usually think, oh, the carbon source is going to be a sugar. It also can be organic acids that the plant is making available. So the plant is feeding the bacteria in exchange for getting usable nitrogen. And it's a good deal for both parties, as long as they both shall live. They'll be in that that mutualism. I like mutualism. It is the best symbiosis. Parasitism is always sad, but mutualism is happy to me. We're going to be done early today, which is all right. So I want to go back to this and kind of put this in a larger context. A lot of the stuff I teach is like, hey guys, this cell, this protein, that's so great. There's always a bigger context too. And for this, this kind of picture, we are like the rabbit. We are like the rabbit. We are the, consumers of the plants that are primary producers that depend on these processes. So what's good for nitrates in the soil is good for plant growth. And what's good for plant growth is good for the organisms that consume plants, like rabbits, or cows, or sheep, or people, or lemurs, or like whatever animal we're talking about. So there's a couple things on here that I haven't mentioned, or haven't mentioned strongly enough. Plants that make nodules have the nitrogen-fixing bacteria with them, giving them nitrogen, but they can also still take up nitrates through their roots like any other plant. So it's like they're getting a double boost of nitrogen intake, partly from their nodule bacteroids and partly from nitrates in the soil. Other plants, and this always looks like marijuana to me, or maybe it's supposed to be like one of those weird Japanese maples, but it's representing a non-leguminous plant, a regular plant. They also depend on nitrate availability in the soil, which comes from the nitrogen fixers together with nitrifying bacteria. Regular plants depend on this stuff too. They just aren't hosting nitrogen fixing bacteria in their root nodules. They don't have any root nodules. Plants are somewhat in competition for nitrate with denitrifying bacteria. And that is kind of an example of a zero-sum game. Like, there's a certain amount of nitrate in the soil. Plants are trying to get it. Denitrifying bacteria are trying to get it. And those would be competing for the same resource, nitrates. The other word on here that I haven't mentioned is assimilation. Assimilation. And that tends to be a word that we use in plant nutrition, but not animal nutrition so much. So plants can uptake and use nitrate. So we called it usable form. When an organism can uptake and use an element to build part of its body, it's called blank assimilation. Assimilation. Certain organisms have to have certain forms of elements that they can work with. So for plants, nitrate is the version of nitrogen that plants can assimilate and actually use. Like this is stupid, but if I, like, oh, my plant needs protein, it needs nitrogen. We don't go like bury some chicken down there at the roots. Like eventually that can break down into and eventually make its way back to nitrate. But what do we put? We put fertilizer, which is mostly ammonium nitrate, which is like a double boost of nitrogen that plants will be able to use. That's what we put. And then our last link in here is the decomposers. So the decomposers are doing something that is also very important in this system. Decomposers can be fungi and bacteria. And what they're doing is they are converting proteins and nucleic acids, for the most part, back into, it's not nitrate, it's ammonium. Back into ammonium. So they are recycling the macromolecules out of the plants, and if the rabbit were to die and just decompose into the soil, the rabbit's proteins and the rabbit's nucleic acids would be broken down by the decomposers, and their waste product is ammonia, which goes back into the soil. And then you have circle of life stuff. And this is part of nitrogen cycle. We'll do a little bit short segments on nitrogen cycle and carbon cycle on Tuesday. So this is kind of ecology and kind of microbiology and kind of plant biology, but these conversions are really essential for all life on Earth. So the kind of stuff that I ask on the test is you'll have this picture, and I will give a scenario. For example, if you suddenly had twice as many nitrifying bacteria in a soil, like that's the scenario, you've got twice as many nitrifying bacteria. The question is always going to be, how does that affect the rabbit? Like, I'm going to make it animal centric because that's how most of us tend to think. How would that affect the rabbit? And then explain your reasoning. And there's probably like 4 or 5 of those kind of questions going with this picture. Twice as many nitrifiers. What would be the result of that on the rabbit? Or stuff like, if the soil just flooded and it stayed that way for 3 weeks, what's the effect on the rabbit? Those kind of questions. If the leguminous plants had a mutation and they can't make a nod factor, what's the effect on the rabbit? So you kind of follow the nutrient path around and think ecologically, and that will be one of the bigger questions next Thursday on the exam. So be working on your unknowns. Don't forget to take pictures of them. Don't forget you need to do 2. You're supposed to do 2 unknowns. And if you have questions about those, just email me. Otherwise, I will see you Tuesday morning.